Hey everyone, and welcome to this explainer. Today we're taking a really fascinating trip into the microscopic world to see exactly how our cells survive under pressure. Whenever a cell encounters a stressor, it basically has two choices. It either has to adapt or it dies. So we're going to strictly examine how cells adapt to stress and injury, and we'll be looking at some direct biological examples to make sense of it all.
Okay, let's dive into this. Here is our highly structured field guide to cellular changes. We'll start with the general forms of adaptation, then we'll break down hyperplasia, hypertrophy, atrophy, metaplasia, and finally, dysplasia.
Section 1. Forms of cellular adaptation. The four main types. Think of cellular stress as the ultimate biological problem. Maybe the environment changes, workloads suddenly spike, or the nutrient supply just drops. Well, to survive, the cell comes up with a structural, biological solution. It fundamentally changes its physical state. Based on our source material, these adaptive responses primarily fall into four main categories: hyperplasia, hypertrophy, atrophy, and metaplasia. Each one is a totally unique response to a very specific set of conditions.
Section 2. Hyperplasia, increasing cell number. Let's move to and see how this builds into our first specific category. So the textbook definition of hyperplasia is an increase in the actual number of cells in an organ or tissue. And naturally, that directly leads to an increased overall size or mass of that tissue. But here is the absolutely crucial operational rule to remember. This specific adaptation can only take place in cells that are actually capable of synthesizing DNA. If a cell can't divide, well, it simply cannot undergo hyperplasia. No way around it.
So how does this actually play out biologically? Well, the mechanisms are honestly beautifully precise. It involves the production of transcription factors that act as triggers for growth. In some cases, like hormonal hyperplasia, the hormones themselves act directly as growth factors. And then there's compensatory hyperplasia, like, how your liver can actually regenerate after a piece is removed. In that case, you get a rapid proliferation of remaining cells, plus brand new cells developing right from stem cells.
And this brilliantly illustrates exactly what we're talking about. This is benign nodular prostatic hyperplasia, or NHP. It's a classic example of pathologic hyperplasia driven by an excess of androgens. Notice how the hyperplastic glands are basically stacked back to back. The sheer number of cells has literally multiplied dramatically because of that intense hormonal stimulation.
Section 3. Hypertrophy, growing in size.
Now, shifting our focus from cell quantity to cell volume, we have hypertrophy. By definition, hypertrophy is an increase in the size of the individual cell. But you really need to understand that this happens due to the increased synthesis of structural components inside the cell. It's an active structural bulking up. It is absolutely not just passive cellular swelling from taking on water. Remember that rule we just established about dividing cells? Right. Well, here is where it dictates the cell's available stress response. Dividing cells can actually undergo both hyperplasia and hypertrophy at the exact same time to meet demands. But non-dividing cells, like the myocardial fibers in your heart, they only have one option. Because they literally cannot divide, they undergo hypertrophy only. And why does this matter? Well, because when heart muscle can't multiply to share a heavy workload, it just has to bulk up, and that carries some major health implications. To really visually cement this concept, just look at how a single cell changes here. When a cell undergoes hypertrophy, it doesn't split or multiply. A normal baseline cell just synthesizes a ton of structural proteins to become a significantly larger, much more robust version of itself, so it can handle that increased functional demand. According to the source material, the triggers for this kind of massive growth fall impartially into two types. First, you have physiological hypertrophy. This happens because of normal increased functional demand or growth factor stimulation. A great example is the massive uterine enlargement that naturally occurs during pregnancy. But then you have pathological hypertrophy. This happens in disease states, like the dangerous thickening of cardiac muscle in the left ventricle when someone has chronic systemic hypertension. Section 4. Atrophy, shrinking cell size. Now we're shifting the narrative slightly to our third category, which is a degenerative adaptation rather than growth. Atrophy is fundamentally a decrease in the size of a body organ, tissue, or individual cell. And this physical shrinking is inextricably linked with a decrease in function. The cell is literally scaling back its operations just to survive. And, this happens for a whole host of stress factors: disease, injury, loss of nerve supply, poor nutrition, or sometimes just a simple lack of use.
So, a cell physically shrinks because atrophy is the result of either decreased protein synthesis or increased protein degradation. The source actually outlines two very specific step-by-step mechanisms for this degradation. First, lysosomal acid hydrolases actively chew up and degrade proteins taken in from the outside. Second, the ubiquitin proteasome pathway flags and degrades a lot of the cell's own internal proteins. Basically, the cell breaks down its own structural components to save energy.
If you direct your attention here, this micrograph really proves the concept of aging-associated tissue loss. What we're looking at is an atrophic testis. There's a marked, highly visible loss of germ cells within the tubules, and it's accompanied by fibrosis around and between them. The tissue has literally regressed physically as the functional demand and support have just waned over time.
Section 5. Metaplasia. Replacing cell types. Okay, moving on to our fourth adaptation, and this is a truly fascinating concept. Cells actually shifting their identities to survive. The exact medical definition of metaplasia is a reversible change where one adult, fully differentiated cell type is completely replaced by a different adult cell type. But the native cells don't just magically morph. Instead, this happens because stem cells in the tissue are actually reprogrammed. This allows a tougher cell type to take over an environment that the original cells just couldn't handle anymore. Now, what's really interesting about this slide is that it shows a section from the cervix displaying squamous metaplasia. Normally, endocervical glands are lined with these very delicate columnar cells. But here, because of chronic irritation or infection, those delicate cells have been entirely replaced by a much sturdier squamous lining to protect the tissue underneath. It's an incredible defense mechanism. However, this remarkable survival mechanism does come with a specific warning directly from the text. It states, "The factors that predispose to metaplasia, if persistent, may eventually lead to induction of cancer in metaplastic epithelium." So, while the adaptation itself is an orderly biological response at first, the chronic stress driving it can absolutely push the tissue way too far.
Section 6. Dysplasia. Disordered cellular growth.
This marks the dangerous transition from orderly adaptation into disordered cellular development. Dysplasia is, well, it's chaotic. It features a complete loss of architectural orientation, meaning cells don't align right anymore. You see cellular pleomorphism, so a total lack of uniformity in cell size and shape. There's accelerated cell proliferation and striking nuclear abnormalities. The nuclei become hyperchromatic, staining super dark, and the ratio of the nucleus to the rest of the cytoplasm goes way up. Taking a look at this micrograph of stratified squamous epithelium, you can visually identify some severe dysplastic changes. Notice the diffuse atypia and the complete loss of normal maturation as you move from the bottom basal layer up to the surface. It's totally chaotic. The nuclei are dark, they're irregular, and that nice orderly arrangement we expect to see in healthy tissue, it's entirely gone.
So the crucial point is this. While early-stage dysplasia and all those adaptations we've talked about today, hypertrophy, hyperplasia, atrophy, and metaplasia, are generally reversible if you just remove the stressor, when exactly does reversible adaptation become an irreversible disease? What happens if that cellular insult persists? Because as our source clearly notes, severe dysplasia is known to progress into carcinoma in situ and ultimately invasive carcinoma. It really leaves us wondering just how perfectly balanced our cellular responses have to be to keep us healthy, doesn't it? Thank you so much for joining me on this deep dive into the catalog of cellular responses.